So what is differentiation? The cells become structurally modified to perform a particular function. The elongation phase will not go back to the meristematic phase. And plasticity is also one of the concepts of development in the case of plant. Ethylene is a gaseous plant growth regulator, which is an inhibitor. It neither belongs to the group 1 nor the group 2 plant growth regulators. Auxin is one such hormone which is present at the coleoptile or the tip of the shoot of the seedling. Hello everyone, a warm welcome to another session on chapter 15, that is plant growth and development. I am Dr. Divya, biology faculty, Vidhyaashram Pre-University College, Mysuru, Temple of Excellence. So in the previous session of this chapter, we had discussed about growth rate, and under growth rate, we understood that the growth rate can be measured and it can be depicted arithmetically as well as geometrically. And we also understood that if the growth rate of a plant, say for example, the elongation or the increase in the length of the plant is considered in an arithmetic manner, then the graph that is obtained is a linear graph. And on the other hand, if we consider the size of the cell and all that, in the case of growth rate in a geometric manner, wherein all the cells participate in the division process, then we get a sigmoid curve or a S-shaped curve. And also we studied about the conditions that are required for a plant to grow, wherein we understood that water, light, gravity, nutrients and temperature, all these play a very important role in the growth of the plant. In today's session, we shall study about three important terms in cell division, which is called as differentiation, dedifferentiation and redifferentiation, and also we shall look into the plant development, that is how the plant develops in a sequential order or in a correct manner. How it develops we shall understand, and we shall also study the important growth regulators in a plant and what is the role of these growth regulators. In short, we shall study, but in the coming sessions in detail we shall continue to study with the growth regulator. So to begin with today's session, we shall first see or understand the term differentiation. So differentiation means the cells are becoming different, the cells are changing into something different. So here the cells which are derived from the shoot meristem, that is the root apical meristem and the shoot apical meristem, as well as the cambium. So all this we have studied in the previous session. So because the actively dividing cells are present in the shoot apical meristem, the root apical meristem, the lateral meristem and also the cambial region, right? So in those regions, what happened? The cells that have been derived, those cells which are present in the SAM, the RAM and the cambium, those who have divided and formed new cells, they actually differentiate and they mature to perform specific functions. They differentiate. Say for example, maybe the lateral meristem, the cells that have divided in the lateral meristem might differentiate into a different kind of cell, so to perform a specific activity. So that particular process is called as differentiation. The cells which have divided, those cells, they undergo a change in their structure in order to perform a specific activity. Such kind of cell division or the formation of such type of cells is called as differentiation process, and this differentiation process actually leads to maturation. So the cells would have divided, they would have undergone elongation, and once they undergo elongation, they undergo maturation. So that particular process is nothing but differentiation. And during differentiation, the cells usually undergo major structural changes both in their cell walls as well as their protoplasm. Why? Because the cells after maturation, of course, they will undergo certain structural changes because of the organelles present in them, because the depositions that occur in the cell walls and also the protoplasm. So when I say protoplasm, protoplasm is also nothing but it is the cytoplasm, along with the nucleus, it forms the protoplasm. So the development of the nuclear membrane, all that will occur properly once the cell matures, right? So therefore, there will be a structural modification in the protoplasm as well. So therefore, it is called as differentiation. I told you differentiation means structurally they are changing into something else. The cells, the meristematic cells which have divided, they are structurally changing in terms of both the cell walls as well as the protoplasm. So the best example that we can take of is the trachary elements. So trachary elements that are present in the xylem. So we know that the xylem, which is a water conducting tissue, it actually is made up of different types of components such as the sieve tubes, the xylem vessels, the trachary elements, and so xylem parenchyma and all that, right? Among them, I'll take the example of the xylem trachary elements. So these xylem trachary elements, once they mature, or once they, in other words, I can say once they differentiate, they completely lose their protoplasm. So this is how a normal cell will look with their protoplasm, that is having the cytoplasm, having the nucleus, and having all the other cytoplasmic contents or the organelles in them. So this is how it looks when they mature. But these mature cells, now they will differentiate. That is, they will completely change their structure. Could you imagine a cell from here now how it is looking? That is the best example we can take of is the trachary element of the xylem. So they will start to become narrower, tapered at both the ends. The shape of the cell completely changed, right? It became narrower, it got tapered at the end. Therefore, cell wall also changed, the structure of the cell wall changed, and also they started to lose protoplasm. Now these cells do not possess any protoplasm in them, and also they start to become very strong because of the modification in the cell wall. They start to become very strong and they start to become elastic, and these lignin and cellulose depositions will start on the cell wall. That is, they are nothing but called as the secondary cell wall depositions. So first depositions were there on the cell wall, again lignin and cellulose started depositing on the cell wall. So now structurally the cell wall is also changing. So this is nothing but differentiation that we can talk about, and the function also changes. So the function now has become specific. So I told you what is differentiation. The cells, the meristematic cells will divide, they will elongate, they will mature, and those matured cells will completely change its structure. In what form? Completely change its structure when it comes to cell wall and protoplasm. So the best example that we took for explaining was the trachary element. So they will change their structure and structure of the cell wall and the protoplasm in order to perform a specific function. So trachary element is present in the xylem. What is the specific function of the xylem? To transport water, right? So that is what happens here. So the in xylem, the one of the component of xylem is the trachary element. So when a cell, when a matured cell gets converted or gets structurally changed into a trachary element of the xylem, it loses its protoplasm, it becomes elongated. Then the cell wall depositions, that is cellulose and lignin depositions on the cell wall will occur. It will become more elasticated and all that will happen, and their main now they will be structurally organized in such a way that they can carry water to long distances under extreme tension. So now we have understood how the mature cell has changed by structurally changing its protoplasm and the cytoplasm by taking the example of a trachary element, that is, which is a component of the xylem, and their main function is to transport water to long distances to different parts of the plant body. So this is about differentiation. Next, talking about the de-differentiation. So de-differentiation, what do we mean by de? Deforestation means you're planting trees again, right? So here, what happens here in this particular case is de-differentiation. Again, differentiation will occur. So what happens, as we know, once the cell becomes mature and they become structurally organized, or they change structurally in order to perform a specific function, they will not revert back to differentiation. It is final. Their structure is fixed. But in the case of de-differentiation, what happens? It is a very interesting that is phenomenon wherein the living differentiated cells that have lost their capacity to differentiate will again start to differentiate, or will again start to divide and again start to modify structurally. That process is called as de-differentiation. De-differentiation means again differentiating. So it is easy to understand. So what happens here? In the previous, I told you the cells were differentiated. Those cells which had differentiated, they for some time they will lose their capacity to differentiate. But after some times, again they regain their capacity to divide and differentiate. Such cells are called as de-differentiated cells, and the phenomenon is called as de-differentiation. The best example that I can take of is the formation of meristems. So here in the formation of meristems such as the interfascicular cambium and the core cambium, they form fully differentiated parenchyma cells. So could we imagine that these cells can differentiate back? So what is differentiation? The cells become structurally modified to perform a particular function. So in this particular case, if we can see the cells here, that is the interfascicular cambium. These are the meristematic cells, having the meristematic cells, that is the interfascicular cambium and the fascicular cambium, the vascular cambium and all that. Those cells have already undergone differentiation, because we know that the vascular cambium, say for example, xylem has already undergone differentiation. And it is performing its specific function of carrying water. But in some cases what happens, they will again change its structure. So that is what happening here in the case of interfascicular cambium. So interfascicular cambium are the cambial cells that are present between two vascular bundles. So this region is the interfascicular cambium. Next we have the fascicular cambium. So fascicular cambium is the cambium cells that are present between the xylem and the phloem, and they actually provide some cells for the formation. Actually it is the vascular cambium cells that actually gets differentiated to form the xylem and the phloem. But in this particular case what happens, now they have differentiated. They can de-differentiate back and they can become parenchyma cells. That is what it means by de-differentiation. So what happened here? The interfascicular cambium, the fascicular, this is I, the fascicular cambium and the vascular cambium, all these again got de-differentiated. They had already become differentiated. Those differentiated cells again got de-differentiated. They structurally modified themselves and they became parenchymatous cells. So that is what is de-differentiation. So next we will move on to redifferentiation. So here the meristem or the tissues, meristem cells or the tissues, they are able to divide and produce cells that once again lose the capacity to divide, but they will mature to perform a specific function. So now the de-differentiated cells once again lose their capacity to divide. They'll stop the division process. The cells that have already divided will mature and they will start to perform specific function. That particular process is called as redifferentiation, and such cells are called as redifferentiated cells. So what happens here? The de-differentiated cells will stop its division process and they will start to mature. Those matured cells will lose their capacity to divide and they will start to perform a specific function. Such type of cells are called as the redifferentiated cells. So now we know the three important terms: differentiation, de-differentiation and redifferentiation. Next, moving on to studying about the development of the plant. So far we understood how growth occurs, what are the phases of growth. So when we were studying about the phases of growth, we understood that there are three phases of growth called as the meristematic phase, then came the elongation phase and then the maturation phase. So this phase actually continues in an orderly manner. The elongation phase will not go back to the meristematic phase. Once the meristematic cells divide, they elongate, and then they reach maturation. Then their final stage is senescence or the death of the plant, but they will not revert back. So that is the development process. So all changes that are taking place in an organism, be it a plant or an animal, here during its life cycle, from the germination of the seeds to senescence, it is in a sequential order. So a plant, once you sow the seeds and once the seedlings come out and the seeds develop into the plant, they mature and they die. Until then, the growth of the plant is in an orderly manner. Therefore, we shall understand the sequence of development of the plant during its life cycle. So what happens here? First, meristematic cells will be there. So what are those meristematic cells? They are the cells which undergo cell division. So they will divide, they will form new daughter cells which are also meristematic, and the old meristematic cells that were there, they will actually undergo plasmatic growth. That is, they will start to, that is, plasmatic growth is nothing but the cell wall depositions and all will occur, cell wall elongation will occur, the formation of the protoplasm, all that will occur, usually happens in the expansion phase or the elongation phase. As I can say, the meristematic cells, they will divide. Now these meristematic cells will again divide. This process is continuous, which will be taking place until the death of the plant. But what about the cell that has already divided? These cells, they will enter into elongation phase. So what happens in the elongation phase? So as you can see here, in the elongation phase, the plasmatic growth will occur. Therefore, expansion of the cell will result in. So the cells will expand. So like this, they will start to expand. And it is in the elongation phase actually the increase in the size of the plant will take place. So that is why imagine a small seedling, how it will develop into a huge tree, how it will increase its size into a huge tree, right? That is during the, because of the expansion of the cells or during the elongation phase. Next, those cells which have elongated. Now elongation will occur. These cells will also start to elongate because they've already finished the division process, right? Now, what happens to the elongated cells? These elongated cells will then enter into the differentiation phase. Differentiation or the maturation phase we can say, or it will enter into the maturation phase. So in maturation phase what happens? Differentiation will occur. Now the elongated cells, they have to structurally modify in themselves into such a way that they can perform a specific function. I explained using the trachary element of xylem in the previous slide. So it is understood. What is the fate of each cell? Means what is, the cell will differentiate itself structurally in terms of cell wall and in terms of protoplasm in such a way that they can perform a specific function. Now they have reached the maturity. Will these mature cells go back to the meristematic phase? No, it will never. It is in a continuous sequential manner like this. After maturation and all that, what happens? It is the senescence stage. So in senescence stage what happens? All the leaves and all that will start to wither. So once the leaves and all wither, what happens? Obviously photosynthetic rate will completely decline in the plant and the plant will die. So that is what happens in the last stage, that is the death of the plant, or it is the senescence stage of the plant. So this is how the plant sequential order they will grow. So now talking about development. In development, there are different terms that we have to concentrate on, and one such is called as the plasticity. So plasticity. So plasticity is nothing but the ability of the plant to look different when compared to that of the juvenile phase or the young phase. So some plants, when you see, say for example the leaf structure, they will look completely different. But when they mature, the leaf structure will look completely different. That is called as plasticity. And plasticity is also one of the concepts of development in the case of plant. So what is plasticity? The ability of plants to follow different pathways in response to environment or phases of life to form different kinds of structure. So, At one period of their growth, they will be having parts of different structure, and in other period of growth, they will have parts of different structure. It would have got modified, and that modification may be because it is one kind of phase of growth of that particular plant, or maybe it is because of the environmental stresses the plant is subjected to. No matter what, the plants usually they exhibit plasticity, that is, from the younger stage of the plant will look something different when compared to the adult or the matured stage of the plant. Now taking example, so this is called as heterophily. So we can use the word heterophily for that. So heterophily, hetero means different, phyll means leaf. So leaves exhibiting different structures in one particular plant is called as heterophily. So heterophily in cotton can be taken as the example, in coriander can be taken as the example, and one more is larkspur. So these leaves we can take the example. So what we can see is, first we will see the heterophily in larkspur. So in larkspur and in buttercup. So in both of these we shall see the heterophily. So if you see the leaf in the younger stage, that is juvenile phase. So I told you plasticity can occur because of environmental factors also, and plasticity can occur because of the phases of growth also. So here we are talking about the plasticity in terms of phases of growth. So here you are seeing heterophily. In one plant itself you are seeing two kinds of leaves. Even coriander is also, so coriander, that is dhania leaf, is also one of the best example for heterophily. If you see the young leaves of coriander, it will be oval like this. And older leaves, when you see the plant, coriander plant, young leaves usually they look like this, right? But older leaves, when you see, it will be somewhat like this, right? So that is why a change. So here itself you can see one kind of leaf and different kind of structure in the leaf. So it is called as heterophily. Heterophyll or heterophily. So here, if you observe the plant larkspur, the young leaves look somewhat like this, whereas the adult leaves look completely different from that of the young leaf. If you observe the margins of the leaf, it is completely different. And likewise, if you look buttercup. So buttercups, based on the environmental or the habitat in which it's growing, it will show or exhibit heterophily. So buttercups, if they are growing in a terrestrial habitat or if they are growing completely on land, then the shape of the leaf is completely different. See here. And if the same buttercup is growing in water-logged area or in water condition, then the leaves look completely different. So this is also heterophily. Heterophyll. So this is about this such a condition is called as plasticity. So almost all the plants exhibit plasticity depending on the habitat in which they are growing and the depending on the phases of its growth. So here the leaves of the juvenile plant, they will be different in shape. So juvenile plant means young plants. They will be different in shape when compared to the mature plants. So here you can see in larkspur, the shape of the leaf of juvenile plant is completely different when compared to that of the adult plant. So the young leaves will have a different shape when compared to that of the adult leaves. Like that also we can say. And on the other hand, difference in shape of leaves produced in air and those produced in water in buttercup. So buttercup is exposed to a terrestrial habitat wherein the leaves are exposed to the air in spite of water. Then the shape of the leaf is different, and when they are completely exposed to water throughout, the shape of the leaf is completely different. So all these means they are heterophilous development of the leaf, which is nothing but plasticity. So plasticity is a mechanism wherein the plants are capable of following different pathways in response to environmental changes or in response to the phases of the life cycle. So that is called as plasticity. So plasticity is development of a plant also exhibits plasticity. So let us talk about the factors that are responsible for the development of the plant, that is factors in development. So when we say factors, there might be some external factors which are called as the extrinsic factors, and some internal factors which are called as the intrinsic factors. So external factors means they are the factors to which the plants are exposed to. That is, they belong to the external environment that is outside the plant body, such as the light, the temperature, water, oxygen, nutrition, etc. So light also plays a very important role in the growth of the plant and development of the plant, right? Because light is one of the main raw materials which is also involved during the process of photosynthesis, wherein plants, they make use of the carbon dioxide and water, and along with the light that is taken in by the chlorophyll, the carbon dioxide and water is converted into glucose. So for that, it is very, very important. And glucose, in turn, it helps in the growth of the plant. Next, talking about temperature. So we all know that an optimum temperature is needed for the proper growth of the plant. A temperature below or above the optimum temperature will hinder the growth of the plant. So that is why temperature also plays a very important role. And next, coming to water. Water is also one of the important raw materials in plants, especially for photosynthesis process. And not just that, plants, in order to maintain the proper shape and size of the cells in a plant, water is very, very important. So therefore, water also plays a very important role in the growth of the plants. Next comes oxygen. So oxygen is also important in plant for respiratory process. So we all know that not all the cells in a plant can participate in the photosynthesis process, right? So there are certain cells in the plants which lack the chlorophyll pigments. So what will they do? So they actually participate in the process of respiration, wherein they make use of oxygen and they break down the glucose molecule. So for that, it is very, very important. And next is nutrition. So the amount of nutrition, be it the macronutrient or the micronutrient that is added to the soil, based on that also the plant growth can be determined. So therefore, these are the important extrinsic factors that play a very important role in the development of the plant. Next, talking about intrinsic factors. So intrinsic factors means within the plant body. They may be intracellular. So intracellular means inside the cell. So inside the cell, it is nothing but genetic. That is because of the genes that are present on the DNA strand or on the chromosomal strand. So a good trait or a good set of genes in the plants will give it a good trait. So therefore helping in the growth of the plant, right? Or it can be intercellular factors. So intercellular factors such as the plant growth regulators, that is the growth hormones in the plant, such as the abscisic acid, the ethylene, the gibberellin, and auxin, cytokinin. So all these growth regulators also play a part in the growth of the plant. So this is how the chemicals such as the plant regulators also play a very important role in the overall development of the plant. Say for example, if I say auxin, they helps in the formation of the root or the root elongation. Then the cytokinin helps in the shoot elongation. So like this, they help in the overall growth of the plant. So these are the different extrinsic, intrinsic factors which help in overall development of the plant. So next, talking about the plant growth regulator. So what are the characteristics of the plant growth regulator? So a plant growth regulator should have certain features that actually favor the plant. So what are the characteristics that they hold in order to say they are a good plant growth regulator? So plant growth regulators are also called as PGRs. That is plant growth regulators, PGRs. So they are very small, they are simple molecules which are made up of different types of chemical composition. So each of these are made up of different types of chemical composition. Say for example, there are different types of growth regulators such as the indole compounds. So auxins and all that are the indole compounds. We have indole 3 acetic acid, that is IAA, which is the best example for an auxin. And then we have certain adenine derivatives such as N6, furfuryl aminopurine, then kinetin. So kinetin nowadays it is called as cytokinin. cytokinin. So all these are there. So about all these in detail, about the main growth regulators in detail we'll study in the coming sessions. But for now, if you know the examples, it is enough. And some of the best growth regulators are the derivatives of carotenoids such as the abscisic acid, abbreviated as ABA. So abscisic acid is usually derived from carotenoids, and carotenoids is one of the important pigments that are present in the plant. We have learnt about that in photosynthesis in higher plants chapter. And next is terpenes. Terpenes are also one of the important regulators in plant, growth regulators, such as the gibberellic acid, which is called as GA3. So gibberellic acid usually helps in increasing the size of the plant, be it the size of the leaf and all that. So they are derived from terpenes. This is gibberellic acid, which is GA3. Next, we have certain growth regulators in the form of gases. That is, they are volatile in nature, such as ethylene, which is called as C2H4. So if you can see here, the important characteristics of the plant growth regulators are they are small in nature, they are simple molecules which are made up of different types of chemical compositions, and they are indole compounds. The best example is indole 3 acetic acid, which is IAA, which is the auxin, and then they are made up of some are of adenine derivatives such as the N6-furfuryl aminopurine, and one more is kinetin, which is called as cytokinin. And the derivatives and some of the compounds are, that is growth regulators, are derivatives of carotenoids such as the abscisic acid, which is called as ABA, and some of them are the derivatives of terpenes such as the gibberellic acid, which is GA3. And not just that, terpenes in plants also help in protecting the plants against the insect attack. So for that also it will help. Next is, some of the growth regulators are volatile in nature. They are in the form of gases, that is ethylene, which is C2H4. We came to know about the different PGRs, or the plant growth regulators, that are there in the plant, some of the common ones. So the plant growth regulators, they can be broadly classified into two groups based on their function. Based on what is their function in the plant body, the plant growth regulators are of two types. So one of the group of PGRs are involved in the growth of the plant. So they are involved in growth promoting activities. So what are growth promoting activities? Cell division, cell elongation, maturation, all that are growth promoting activities, right? Such as they help in cell division, then they help in the enlargement of the cell, that is the elongation of the cell, and they help in the pattern formation of the cell, that is how the cells has to be arranged. All that pattern formation they will help, and they will help in the tropic growth of the plant, and they will also help in the flowering in the plant, the fruiting in the plant, and the seed setting in the plant, for all those growth regulators. So those plant growth regulators which are involved in the growth of the plant are put under group 1. So one group, I told you, based on the role that they play in the plant, they are of two groups. So one group of plant growth regulators, they mainly focus on increasing the growth of the plant, such as in terms of cell division, in terms of cell elongation and enlargement, in terms of flowering, seed formation, fruiting, then in terms of that is growth, tropic growth of the plant, and all that. And the best example is auxin, gibberellins, and cytokinins. These are the best plant growth regulators which actually concentrate on or play an important role in the plant, helping the plant to increase their growth. That is auxin, gibberellin, and cytokinin. And the other group of plant growth regulators, they play a very important role in response to wound and environmental stresses in the plants. That is environmental stresses such as biotic stresses and abiotic stresses. I told you terpene previously, that plant derivative from terpene and all that, they actually help the plant in against insects. They protect the plant against insects. So biotic origin means they keep the plant or protect the plant against insect attack, pathogen attack, and all that. They are called as biotic. Why? Because they protect the plant against the insects, the animal, so they also protect the plants against the animals. So some of the animals, if they come to feed on these that is herbaceous plants, sometimes the smell that is given out by these growth regulators or the plant growth regulators itself, they will keep the animals away from feeding on the plant. So that is the reason. Next is abiotic origin, means against all the environmental stresses. And not just that, they also help in the various growth of the plant by inhibiting dormancy. So what is this dormancy? Dormancy is a condition in which the seeds will fail to germinate. So sometimes what happens, because of the environmental stresses the plant is subjected to or the seed is subjected to, the seed will not germinate and develop into a plant. They will remain in an inactive state or in a dormant stage. So such seeds are called as dormant seeds. So in order to break the dormancy in such seeds so that they can germinate, these group 2 plant growth regulators will actually help. And abscisic acid is one such plant growth regulator that belongs to group 2. So those plant regulators which only focuses on the growth of the plant are cytokinin, indole acetic acid, that is auxin, and gibberellin. And those plant regulators that focus on healing of the plant against wounds, against the environmental stresses, and they also help in breaking the dormancy of the seed. The best example is abscisic acid. So, and apart from this, there are also gaseous plant growth regulators, right? That is gaseous PGR. The best example that we can take is of ethylene. So, these ethylene, they actually help in fruit ripening and all that. So, they do not fit to group 1 nor group 2 because they don't help in the growth, nor do they help in the protecting the plant against insects, pathogens, against environmental stresses and all that. But their main function is they can inhibit the growth activity. So ethylene can inhibit the growth activity. That is why, apart from that, ethylene is of great economic importance to the fruit growers. Why? Because ethylene is one such hormone that will help in ripening the fruit fast. So that is why it can be used for economic purpose. But other than that, it is a growth inhibitor. So it inhibits the growth activities of the other hormones. So that is why they have not placed ethylene in neither group 1 nor group 2 of the plant growth regulators. So these are the different types of plant growth regulators and their characteristics in general. So next, talking about the discovery of the plant growth regulator. What exactly led to the discovery of the plant growth regulator? So the first plant growth regulator was discovered by Charles Darwin and his son Francis Darwin. So what they did was they observed that the coleoptiles. So what are coleoptiles? The shoot tips are covered by a structure called as the coleotypes. So coleoptiles are those structures that are present at the tip of the shoot, which actually protects the tip of the shoot. Like how in roots they have the root cap, right? Because in the tip of the root meristematic cells are present. In order to protect those fragile tips of the root caps are there. Likewise, in shoot tip also lots of meristematic cells are present and they are very young and fragile. So in order to protect them, coleoptiles will be present. So Charles Darwin and his son Francis Darwin, they observed that the coleoptiles of canary grass, they responded to, and his son Francis Darwin were the first to observe that there is some chemical component which is actually making the plant bend towards the light. So we all know that the plant grows towards the direction where light is there. And why is it growing towards the direction or bending towards the direction where light is there? Maybe in the coleoptile or the tip of the shoot, there is some chemical compound that is present which is actually being attracted towards the light, so that it is making use of the light and it is helping in the growth of the plant towards that particular light. And that particular phenomenon is called as phototropism. And after a series of experiments performed by them, they found out was in the tip of the coleoptile, there is some chemical compound that is present that actually caused the bending of the entire coleoptile. So they didn't know what it was exactly. They didn't name it as auxin or anything, but they just knew that there is something present at the tip of the coleoptile which is making the plant to exactly bend towards the source of light. Of course, plant will bend towards the source of light because they need sunlight for its photosynthetic purpose, right? So the cells are dividing in that particular direction and growing in that particular direction. What is actually making them grow in such a way? There is some chemical compound that might be present. So such an idea was got by Charles Darwin and son Francis when they were studying or looking into the canary grass, that is the coleoptile tip of the canary grass. And next, one more scientist, that is, his name is F.W. Wendt. So F.W. Wendt was the first scientist to name it as auxin. He was the first to say that there is a chemical component called auxin present at the tip of the coleoptile that actually helps in the growth of the particular plant. So for that, he performed an experiment. So what he did was, in this particular slide you can see, what he did was, he took the Avena species of grass, he took that is the seedlings of that grass he took, and he started studying the shoot region of that particular seedling. That is from the shoot that arised from that particular seedling. He started to study and he started to observe the coleoptile of that particular seedling. So when he observed, he noticed that there is something that is present at the tip of the coleoptile that might actually help in increasing the growth of that particular coleoptile region or the shoot region of the plant. What is it? Is there something present or not? To understand that, he conducted an experiment. What he did was, he took the coleoptile of the that is the shoot region of the plant, that is the seedling, along with the coleoptile. He took it and he cut the tip of the coleoptile. He completely cut the tip of the coleoptile. And next what he did was, he took a agar block and he covered the tip of the coleoptile. So after a few days, he noticed that in spite of covering it with the agar block and in the absence of the coleoptile, the shoot tip did not grow at all. The shoot did not grow at all. It had stopped its growth there. Later on, what he did was, he again removed this agar block and he put that agar block on the intact plant, coleoptile. So one more shoot he took, and without that, wherein he did not cut the coleoptile. On that shoot tip having the coleoptile, so you can see here, the coleoptile region is still present. Coleoptile is still present. He put the agar block on that, and after few days what he did was, he removed that agar block and he put it on the cut tip again. Now he noticed that the shoot tip was growing, and that is when he found out that something from the tip of the coleoptile got transmitted into the agar block, and that agar block, when it was put on a cut coleoptile, it started to grow properly. That is when he understood it is nothing but auxin, and he found out that auxin is one such hormone which is present at the coleoptile or the tip of the shoot of the seedling. So Avena species, that is the oat seedling. So that is how his experiment, it is called as the Wendt's experiment. So it was performed by F.W. Wendt, wherein he was the first person to find out the growth regulator called auxin through his particular experiment on Avena species, that is on the seedlings of the oats. So next is the discovery of gibberellic acid, which is also one of the important plant growth regulator that helps in the increasing the growth of the plant. So we all know about a disease which is called as the Buchanan disease of rice seedlings, and the causative organism of that particular disease is the pathogen Gibberella fujikuri, which is a fungus. So the fungus Gibberella fujikuri, it infects the rice seedling and it causes the Buchanan disease. So how was this found out? It was reported by one scientist called as E. Kurosawa. So E. Kurosawa, in the year 1926, when he was studying for the first time, he was the first to report the appearance of some symptoms on the rice seedlings. And when what he did was, he took the rice seedlings and he treated the rice seedlings with the sterile Gibberella fugiculae. At that time, he noticed that is with sterile filtrates of the fungi. He took a beaker, in that he put water. To that water, he put the Gibberella fugiculae fungi. Now he made a filtrate of that particular water solution. That solution he sprayed it on the healthy rice seedlings. And after few days, he started to notice symptoms development. That is the Buchanan disease symptom development of the rice seedlings. And that is when he first found out that Gibberella fugiculae is the one that actually causes the Buchanan disease or the foolish seedling of rice. So what Kurosawa did was he took a beaker or a conical flask like this, and he took a water solution, sterile water solution. To that, he added this particular fungi, that is Gibberella fugiculae fungi he added, and he made a filtrate of that. That filtrate, he took it and he sprayed it on the rice seedling. On the rice seedling, he sprayed them. So later on, when he sprayed them, he noticed that symptoms started to develop on the rice seedling. So what are the symptoms of Buchanan disease or the foolish seedling rice disease? That we should know first. The first main symptom is paling of the rice plant. The rice plant will become pale, and not just that, the rice plant will increase in height in such a way that it will increase in its height than the normal height. So he was curious, what is this actually increasing the height? That is when later on it was discovered that it is gibberellic acid that is produced by the fungi, that is Gibberella fugiculae, which causes the increase in the height of the rice seedling. And that was found out when they were studying on the Buchanan disease of the rice. So this is how the gibberellic acid was discovered by Kurosawa. Next is the kinetin. So kinetin was discovered by F. Skoog and his co-workers. So F. Skoog and his co-workers, they discovered that in the internodal segments of the tobacco plant, that is the tobacco stem, a callus is nothing but the growth of cells in an uncontrolled manner. So if usually the cells are dividing like this, in callus the cells will start to divide in an uncontrolled manner, and callus usually looks like this. So in tissue culture and all, we get this callus. So callus usually looks like this. So when F. Skoog and his co-workers were studying on the internodal region of the tobacco stem, they found out some callus-like appearance, which is nothing but a mass of undifferentiated cells. So these cells undergo continuous division, but they will not undergo differentiation. What is meant by they will not undergo differentiation? They will not undergo any structural modification. Just they will look like a mass of cells, just like how tumors and all look, just like that. So it is called as callus. And they found out that the division of the cell or the callus formation in that particular plant increased or it proliferated when auxin was added to the nutrient media. So what they did, they took a tube or a tissue culture bottle, then they put the tobacco having the internode. So this is the internodal region of the tobacco, the internodal region. And what they found out was, when they put it in a nutrient media, into that nutrient media they had added auxin as well. So they took a nutrient media like this and they added auxin as well to that. So what they found out was, in the presence of auxin in the nutrient media, and also they had added some extra supplements such as the extracts of vascular tissues, then the yeast extract, then coconut milk or DNA, all these were supplemented to this nutrient media. That is when they found out some callus-like structural development, and the callus-like structural development proliferated when extra supplementary materials, or increased when extra supplementary materials were added to the nutrient media. And later on, Miller et al. In the year 1955, he later identified some crystallized substances and tried to identify what it was. And that is when he found out that it was cytokinins and he named them as kinetin. So now it is called as cytokinin, but earlier those crystallized form, they had called it as the kinetin. So what happened here? F. Skoog and co-workers were doing some tissue culture on the tobacco, that is the stem of the tobacco. So when they put it in a nutrient media along with auxin, they found out that they could see that in the internodal stem region of the tobacco, internodal segments of the tobacco stem, they could find some white mass of cells or group of cells which is called as callus. What they did was they again supplemented that particular media. Again, they put some extra substrates such as the extracts of vascular tissues, yeast extract and coconut milk and all that. And then they noticed that the callus growth or development proliferated or it increased. So they knew about something is actually increasing that particular division of the cells, that is, or increasing the number of the undifferentiated cell. What is that? They didn't know. But later on in the year 1955, Miller et al., he took those structures, that is callus, and what compound was there in that callus, he crystallized them, because crystallization is very, very important in the case of chemistry to identify what is that compound exactly. So that is needed. So he crystallized that particular compound and he named it as kinetin. He said that it was because of kinetin. So therefore, kinetin or auxin, it is one such compound that actually helps in the proliferation of the cells or callus development. Auxin actually helps, and callus development is very, very important in tissue culture. So one small cell of the callus, if you take and put it in a proper nutrient media, you can generate a new plant out of that. So imagine if thousands of callus cells are there in that mass, thousands of plants can be got through tissue culture. So auxin is very, very important hormone in that case. And not just that, when they found out, they named it as kinetin, which is nothing but cytokinin. So along with auxin, cytokinin also helps in the callus formation. It is very, very important. So next important hormone was abscisic acid. So abscisic acid was discovered during the mid-1960s. So abscisic acid, it belongs to the group 2 plant growth regulators, wherein their main function is to protect the plant against abiotic and biotic stresses, right? We know about that. They don't help in the growth of the plant. But so during the mid-1960s, 3 researchers, they actually reported the purification and chemical characterization of 3 different kinds of inhibitors, that is inhibitor B, Abscission 2 and Dormin. They named these inhibitors are because they don't help in the growth of the plant. They are called as inhibitors. So abscisic acid in one way is also a kind of inhibitor. How was it discovered? It was discovered in mid-1960s. 3 scientists, they were working on different forms they were working on, and that is when they found out some chemical compound which they isolated, they characterized and identified them as inhibitors called as inhibitor B, abscission 2 and Dormin. Later on, these 3 compounds proved to be chemically identical. So 3 scientists, though which compound they gave different names, people started to understand that the 3 compounds were of same structure. So that is when it was named as abscisic acid. So actually the 3 scientists' discovery led to the discovery of the abscisic acid. Next is ethylene. Ethylene is a gaseous plant growth regulator, which is a inhibitor. It neither belongs to the group 1 nor the group 2 plant growth regulators. So in 1910, H.H. Cousins, a scientist named H.H. Cousins, they confirmed the release of a volatile substance. So volatile substance means they are capable of evaporating, because ethylene is a gaseous compound. It is volatile in nature. It is capable of evaporating. So they found out the release of a volatile compound from ripened oranges. And not just that, when those fruits were stored, that ethylene that was present in that particular orange, it further increased the ripening of that particular orange. That is when they found out that it was identified as ethylene, which is a gaseous plant growth regulator. So that is how it led to the discovery of the ethylene plant growth regulator. So this was about today's session, wherein we studied the 3 important terms in cell division, which is called as differentiation, dedifferentiation and redifferentiation. And also we understood the that plant growth or development is always in a sequential manner, and we also understood the discovery of the plant growth regulators. We talked about the plant growth regulators in general, the characteristics of the plant growth regulators, and what led to the discovery of the plant growth regulators, how each of the plant growth regulators, such as the group 1 plant growth regulators such as the auxin, the gibberellin and the cytokinin, how they were discovered, and also the group 2 plant regulator abscisic acid, how it was discovered, and the plant growth regulator which is in the gaseous form that neither belongs to group 1 nor group 2, that is ethylene, how it was discovered, all that we have studied. So in the coming session, we shall discuss about the role of each of these plant growth regulators in detail. So we shall meet again in the coming session. Thank you.